All right, so chapter 23. Look, we've talked about the metabolism of sugars quite a bit in this class, haven't we? Haven't we, right? We talked about um, glycolysis, and we talked about the fate of pyruvate as it enters the citric acid cycle. Well, if you look at the amount of energy or consider the amount of energy that is produced when we consume carbohydrates, proteins, or fats, we actually get more than double the amount of energy from the full metabolism of a fat than we do for a protein or for a sugar, for a carbohydrate. And that's why we have to take a look at fatty acid metabolism. Now, if you're wondering, well, I don't consume fatty acids, I consume triglycerides. Well, what we're going to see is that we're going to hydrolyze a triglyceride to produce a fatty acid. So in section 23.1 of this chapter, we're going to talk about how triglycerides are digested and absorbed into the bloodstream. And then in section 23.2, we're going to talk about the concept of beta oxidation, and we're going to explain and we're going to actually quantify why we get so much energy from fats, right? Why is it that we get double the amount of energy from a fat, right, from a fatty acid or from a triglyceride than we would from a sugar? Okay, it's a really good question. Now, again, we know that sugars are a more abundant or a faster source of energy. However, we can get more energy from lipids. So let's talk about lipid metabolism. So I'm going to refer to triglycerides as TGLs once in a while this evening. So what happens when you eat some fat, right? Mr. Dion is a real cheeseburger kind of guy. I had a cheeseburger on my birthday a couple of weeks ago, so I consumed plenty of fat when I ate that. So what happens when we consume fats? Well, fats are not broken up. They are not digested in the mouth right away, right? We have to emulsify fat droplets, okay? It says here that triglycerides, so taken in by fats, are emulsified into tiny fat droplets in the intestine by bile salts from the gallbladder. So you have to understand the concept of hydrophobic and hydrophilic in order to understand how bile serves to emulsify or break apart fat droplets, right? When you consume a fat from, let's say, a cheeseburger, the droplets of fat that you're taking in are pretty big. Okay, so the surface area is not going to be big enough, or there's not going to be enough surface area for the enzymes like pancreatic lipase to act on those fat droplets, and so we have to break them up. Okay, and again, they're emulsified by bile salts. Now, what's bile? I have some figures that show a little bit more information about bile, but if you remember the concept of a micelle when we studied soaps earlier on, when we did the chapter on carboxylic acids and their derivatives, I did a whole section about micelles and talked about the hydrophobic tail and the polar head. Well, instead of using a carboxylate, we're going to make micelles of a molecule called lecithin, and I have a figure of that I took from the book, which is coming up in a second. Also, molecules like cholesterol. If you look at the molecule of cholesterol, it has a polar portion and it also has a hydrophobic portion. Proteins, bile salts. I'll show you the structure of what a bile salt is. Bile salts are derived from cholesterol, and then we have other things like inorganic ions and bile pigments. I'm not so worried about those, but the idea is that we kind of have a whole plethora of different types of molecules, and all they do is enable us to break up those fat globules into something that's smaller. Okay, once we break them up into something smaller, these little tiny droplets, pancreatic lipase, which you can imagine is a catalyst, or sorry, an enzyme that is going to catalyze the hydrolysis of lipids. It hydrolyzes those triglycerides into monoglycerides. All right, we make a monoglyceride and two fatty acids, and I'm going to show you a figure of that as well. It gets absorbed by the intestinal epithelial cells, gets reassembled into triglycerides, which is really strange, and then gets combined with protein to form a chylomicron, which we talked about earlier on in the class. And then we put the triglycerides into the bloodstream where they can get taken up into cells and stored in an adipocyte, which is a fat cell. Well, again, once those triglycerides, the TGLs, once they get into your blood, they get hydrolyzed once again back into glycerol and a fatty acid. The fatty acids are taken up by cells and they're stored in a fat cell, an adipocyte. And you see that in the adipocyte that I have here on the slide, you see that most of it is the fat reservoir, right? You see the little mitochondria that are surrounding the fat reservoir. So if you need energy, what are you going to do? You can extract it from the fat cell, right? We can take those fatty acids and we can convert them into ATP. And that's what we're going to look at in section 23.2. And if we don't need it, well, we're going to store the fats in the fat cells, and we're just going to hang on to it until we need it. Well, let's take a look at what a micelle is. And again, I talked about micelles with you way back in the beginning of the class when we talked about fatty acids, and I talked about soaps. So if you want more information on a micelle, you could look at the look at soaps and how the action of a soap works. And yeah, we even have a lab in our lab manual that talks about soaps and how to make a soap, right? Well, if we look at this molecule, lecithin, so I don't ask you to memorize the structure of lecithin, but you can see that it's a phospholipid. Look, you've got the glycerol backbone here. You've got two fatty acid residues on here, but then you have this phosphate, and then you have this protonated amine here. So that is lecithin. That's the molecule that's shown here. So what's being represented here is all of these purple spheres. All those represent is this portion of the molecule, okay, which is the polar portion. And then the fatty acid chains, so represented by these R groups here, these are the long, brown lines going in. So we have the hydrophilic portion, which again is this polar head group here. And then we have the hydrophobic portion, which would be the fatty acid tails. So the fatty acid tails spontaneously arrange themselves so that they go in towards the center, right? They're going to be repelled by the water molecules, and that's what these are here. They're water molecules, whereas the hydrophilic portion, right, the spheres, those represent the heads of the phospholipids, and those are going to be facing out towards the water, the aqueous environment. Okay, so that's the concept of a micelle. So if we look at the structure of a bile salt, I told you that a bile salt is nothing more than derivative of cholesterol. And I told you that cholesterol is based off of the steroid skeleton, which we looked at. It has the A, B, C, D ring structure. So we have A, B, C, and D. Whenever you see this skeleton in an organic molecule, my friend, you are dealing with some kind of steroid. Now, it doesn't necessarily have to be an illegal anabolic steroid. It could be something like cholesterol, or it could be a bile salt. And these bile salts, again, are made from cholesterol. You can't see the side, and if I start writing, it's going to cover up these molecules on the side. But what I want to show you here is that cholate and chenodeoxycholate, they have polar portions to the molecules, right? They have all these hydroxyl groups, and they both have the carboxylate. Again, I'm kind of losing it here, but they both have the carboxylate. But then they also have the big carbon skeleton, right? That big carbon skeleton is going to be hydrophobic, and the hydroxyl groups and the carboxylate, those are going to be hydrophilic. And the idea is that we want to emulsify fats which are hydrophobic in an aqueous environment so that we can act upon them with a lipase. So here's a figure. I was doing a search in some other books that I had, and I found this in a book that I had, and then I found it on the internet. I can't remember if this comes from an old version of our book, but I don't think this figure is in our textbook. But this is, it looks really simple, but it's a good way of explaining the digestion of a lipid. So we have our fat droplets that we take in. They're really big, right? You can see they're pretty big. But then we have the micelles of bile salts, right? Cholesterol, lecithin. And all we're doing is we're emulsifying. And you see that the fat droplets here, you see they're smaller. So what happens when we take, something that's big and we break it up into a bunch of smaller portions? We're going to increase the surface area, aren't we? We're going to have a much larger surface area, right? Let's say you have a basketball, okay? Yeah, and you measure the surface area of a basketball. Let's say you have an equal volume of peas, right? The kind of peas that you eat, okay? If you have an equal volume of peas as the volume of the basketball, which one's going to have more surface area? Well, it's the volume of the peas, right? Because you have to take into account each tiny little sphere. So all we're doing is we're increasing the surface area, and then it can be acted upon by a lipase, which breaks it apart into fatty acids and into monoglycerides, and that is what makes the micelle. All right? So let's talk about the emulsification. Here you have the giant fat globule, if you will. Okay, it's really big. The fat globule gets broken up and coated by lecithin and other bile salts. Now, I know this is kind of might seem like a funny way to represent the bile salt, but you'll have to take it the way it is. You have the lecithin that's got the polar head group. You've got the hydrophobic tails. Let's just say that the bile salt, I've always laughed at the way this is represented, but it's good enough. It definitely gets the point across. You've got a hydrophobic portion, and you got a hydrophilic portion and a hydrophobic portion, right? So the hydrophobic portion is going to be the one that's going to come into contact with the lipid, and then you have the hydrophilic portion on the outside that's going to interact with the aqueous environment. So there you go. Now we've broken up our fat globule into these little tiny droplets. Then what's going to happen is those little droplets are going to get acted upon by an enzyme called pancreatic lipase. Now, again, this has always been kind of puzzling to me, but it's the way nature designed it, is that you take your triglyceride here, and the pancreatic lipase usually only clips off 2 of the fatty acid chains and makes what's called a monoglyceride. And it's usually the middle fatty acid that's left over. I don't think it's like that 100% of the time. Even in our book, it says it's usually that way, okay? But now you've broken it apart, okay? What have you taken? You've taken your fat, you've emulsified it, which allowed pancreatic lipase to act upon it. Now what have you got? You've got a monoglyceride and a couple of free fatty acids. Well, let's see what happens to those. Those are going to combine with other molecules like cholesterol, okay? I'm going to skip this slide and I'll come back to it in a second. Those are going to combine with other molecules like cholesterol, vitamins. That's not really neither here nor there for us, but the whole idea is that they get combined into my cells, okay, that are going to be coated with the bile salts, and then we're ready to go for absorption in chylomicron formation and absorption. So before we get into chylomicron formation and uptake, let's take a look at the action of pancreatic lipase. This is kind of a review of the enzymes chapter, right? Everybody who's hearing the sound of my voice knows that if you have a lipase, it's going to act upon a lipid, right? So I hope that you all remember the structure of a triglyceride. We have the glycerol backbone. You have 3 fatty acids attached to that backbone. But now the pancreatic lipase, again, like I said, is kind of interesting in that it only breaks 2 of the ester bonds, and you're left over with a monoglyceride, which is a new molecule. We've never looked at a monoglyceride until just now. And then you end up with the free fatty acids. All right. All right, let's move on from there. So again, this is where it gets really interesting to me. This is something I've always found interesting, is that the monoglycerides that we formed in the fatty acids are reassembled into triglycerides that are combined with protein to produce the lipoprotein that we call a chylomicron. So you see that everything is all put back together, reassembled to form the triglyceride and make the chylomicron. So the chylomicron is just a collection of lipids and proteins, and it's going to be secreted into small lymphatic vessels that eventually arrive in the bloodstream. So we have the chylomicron. It arrives in the bloodstream, and then once it's in the bloodstream again, the triglycerides are hydrolyzed back to produce glycerol and free fatty acids, and then they get absorbed by cells. So again, we make the adipocyte, and if the body needs energy, the fatty acids are degraded to ATP, and if we don't, we store them, right? So there you go. That's the whole concept of taking up lipids. So again, the fatty acids are stored in the form of triglycerides. Most of the triglycerides in our molecule are stored as fat droplets in the cytoplasm of adipocytes, or fat cells that make up adipose tissue, which is our fat tissue. And each adipocyte contains a big fat droplet, as I showed you a few slides ago, that accounts for most of the volume of the cell. If you look at other cells, like say those in the cardiac muscle, for example, they have very few fat droplets inside of them. Okay, so in the adipocyte, the fat molecules, or the fat droplets, are surrounded by mitochondria. So again, when we need the energy, the mitochondria are going to act upon those, and the fatty acids are going to be completely oxidized, and then ATP is going to be produced. And before we get into section 23.2, I just want to remind you that the fatty acids provided by the hydrolysis of triglycerides are a really rich energy source for the body. The complete oxidation of fatty acids releases a lot more energy than the oxidation of a comparable amount of glycogen. So let's take a look at the last section. So you've made it this far in Chemistry 102. This is the last section that we look at in the book, section 23.2, which it deals with fatty acid degradation. So there's an interesting experiment that was performed by a chemist named Franz Knoop a long time ago. He was a German scientist, and he did an experiment where he took what are called omega labeled amino acids, and then after they were metabolized, he analyzed what was left over from these omega labeled fatty acids. Now, here's what he did. Now, this is really ingenious. Now, nowadays, we wouldn't do an experiment like this because we have radioactive elements that we can incorporate into molecules that enable us to trace them throughout the human body. But back then, he designed a really ingenious experiment. And here's what all he did was he took an omega labeled fatty acid. And the omega labeled, all it means is he put a phenyl at the very end of the fatty acid instead of just a CH3 group. And when he had an even number of carbon atoms in the chain, he noticed that he ended up with acetate and phenylacetate. But when he had an odd number of carbons, he didn't end up with the same molecules. He ended up with acetate and benzoate. All you need to know about this is that his conclusion is that the fatty acids were oxidized or metabolized 2 carbons at a time, because no matter what, he ended up with these 2 carbon units. Okay, and if he didn't have 2 carbons left over, then the metabolism wouldn't continue. So that's the whole point of this experiment. We now know that the 2 carbon fragments produced by the degradation of fatty acids are not the acetate molecules shown here, but acetyl coenzyme A. And the pathway for the breakdown of fatty acids into acetyl coenzyme A, that's what we call beta oxidation. So if you remember when we talked about glycolysis and the citric acid cycle, we looked at all the different steps that occur. Well, we're going to look at the steps of beta oxidation, but this is the last stepwise pathway that you're going to look at in Chemistry 102. And I promise it's a lot shorter than the other pathways that we look at. And I think it will be abundantly clear to you how it works when we get to the end of it. So beta oxidation, steps 2 through 5, are a set of 4 reactions whose overall form is similar, close to the last 4 reactions of the citric acid cycle. Each trip through the sequence of reactions releases acetyl coenzyme A and returns a fatty acyl coenzyme A molecule that's got 2 less carbons. So we end up producing reduced coenzymes. We make 1 molecule of FADH2, which is equal to 2 ATP molecules, and 1 molecule of NADH that's equivalent to 3 ATP molecules every time we do the beta oxidation cycle. Now you notice that I skipped step 1. Step 1 is what we call the activation step. And step 1 is just like the start of glycolysis, in that we have to invest ATP in order to kick the cycle off, in order to kick off the reactions of beta oxidation. So here's an overview. If you take a look at all the steps, it's a little fuzzy here, but you can see that we end up taking our fatty acid molecule and we end up removing 2 carbons in the end that are incorporated into acetyl coenzyme A molecule, which is going to enter the citric acid cycle. But along the way, we produce 2 ATPs from an FADH2, and we also produce 3 ATPs for an NADH. Now remember, this slide only represents 1 cycle of beta oxidation. For every 2 carbons that we remove, we're undergoing a beta oxidation. Now, if you're not 100% sure what I mean by that, again, I promise that once we take a look at an example, it will become abundantly clear to you. So let's take a look at all the different steps here. Again, the first step is the activation step. The enzymes that we use for beta oxidation are located inside the matrix space of the mitochondria. In order to get the fatty acid molecule, or the acetyl coenzyme A molecule, which comes from the fatty acid molecule, in order to get it into the matrix, we have to use a special transport mechanism to get it into the mitochondrial matrix. And we'll talk about that in a second, very quickly. But step one is just that activation reaction that produces a fatty acyl coenzyme A molecule. Now, that's different than just a plain old acetyl coenzyme A molecule, right? What we're doing is we're taking our fatty acid, which could have, any number of carbons. I mean, it's not going to be unlimited, but we have, let's say, a large number of carbons in our fatty acid. During the activation step, we have the reaction between coenzyme A, the fatty acid, and ATP. And here we actually end up using 2 molecules of ATP for the activation step. It's not written here, but we end up using the value of 2 ATPs, right? Because we end up with adenosine monophosphate and the 2 inorganic phosphates attached together like this. So all you really need to know is that you have an enzyme called acyl coenzyme A ligase, and you end up binding coenzyme A, which we usually represent like this, right? We put the thiol and coenzyme A, and you make a thioester bond. And that thioester bond, this bond here, is a high energy bond. So all you've done is taken the fatty acid that came from your hamburger triglyceride, and you activated it into a fatty acyl coenzyme A. Again, don't get that confused with acetyl coenzyme A, right? Acetyl coenzyme A, you only have the 2 carbon atoms here. 2 carbon atoms, that's acetyl coenzyme A. When we have the whole fatty acid chain here, that's fatty acyl coenzyme A. Now, the whole concept of crossing into the matrix space, I've never asked a student a question about this on an exam, so we'll kind of take a look at it quickly. But the whole idea is that we have to use these carrier molecules in order to get the fatty acyl coenzyme A molecules into the matrix space. And you need a couple of enzymes to enable this to happen. So the first one is carnitine acyltransferase 1. And what that does is it causes a reaction between a molecule called carnitine. We'll look at the structure on the next slide, and acyl coenzyme A, and it forms what's called acylcarnitine. And then acylcarnitine is able to cross into the intermembrane space and then into the mitochondrial matrix by the aid of translocase, what's called translocase. So if we take a look here, here's just the activation or the formation of acylcarnitine. So this is carnitine here. I'm not going to ask you the structure of carnitine. It's just a molecule that attaches itself to the acyl coenzyme A molecule. You make the acylcarnitine, and here is a figure that comes straight from our book. It's very self-explanatory, I think. Here's the activation of, or the reaction, if you will, between carnitine and acyl coenzyme A to form the acylcarnitine molecule. Then that can get transported into the mitochondrial matrix through the translocase, which is here, and then you have another molecule that basically rips off the carnitine, and that's carnitine acyltransferase 2. So I don't know what I would really ask you specifically about this. Nothing more really than in order to cross, in order for the fatty acid molecule to essentially cross into the matrix, it has to have some help from these enzymes, right? The transferase and the carnitine acyltransferase 1 and 2 and the translocase. Nothing more than that. All right, well, let's get into the good parts. And the good parts, I think, or the most exciting parts, are steps 2 through 5, because this is the demonstration of how we obtain so much energy from a fatty acid. So it says here, step 2, and we're going to look at steps 2 through 5. So 2, 3, 4, and 5. So this step is an oxidation reaction. What do you know about oxidation reactions in organic chemistry? They involve either the gain of oxygen or the loss of hydrogen. And what we end up doing is, you see these 2 carbons here, we end up removing 1 hydrogen from each of those to make a double bond, right? It looks like an alkene. Technically, it's not exactly an alkene, but it looks like an alkene, and that would suffice for this class, okay? So if the fatty acyl coenzyme A molecule is undergoing oxidation, right? This is oxidation. Reduction and oxidation are simultaneous, right? They have to occur together. They are not mutually exclusive. And so FAD gets reduced to FADH2. Well, we know that FADH2 has the value of 2 ATP molecules. So again, and I'm just going to stop here quickly, that means that every 2 carbons that we can rip off of our fatty acid molecule, we can produce an FADH2. Well, what happens next is basically a hydration reaction where we add water across the double bond. Okay, so we add water across the double bond and we end up hydroxylating the beta carbon. So here's our alpha carbon, here's our beta carbon. We have the beta hydroxyl of our fatty acyl coenzyme A molecule. And the enzyme that catalyzes the reaction is enoyl coenzyme A hydrase. I'm not going to ask you about that. It's just a hydration reaction, nothing more than that. Okay, so now you've got that hydroxylated beta carbon. It gets dehydrogenated, or you can see that it's getting oxidized, isn't it? Right? You see you have the alcohol, what looks like an alcohol is being oxidized to a ketone. Well, if we have oxidation, then we have to have reduction occurring as well. And when this oxidation occurs, we have the NAD plus being reduced to NADH. And you know that every NADH molecule has the value of 3 ATPs. Okay? Well, we've already made it to the last step. And this is probably the coolest step in the whole entire thing, because not only did we harvest energy in the form of the FADH and the FADH2 and the NADH, well, now we're just going to rip 2 carbons off of that whole giant chain, and we're going to do the whole process all over again, right? If you draw the coenzyme A, and just forgive me here, I'm going to zoom in a little bit. If you draw the thiol here, right, you can kind of think about a bond being formed between that sulfur and this carbon right here. So what have we done? We've removed those 2 carbons that I have in that red circle. They came from the fatty acid, right? That's where they came from. Well, hold on. We still have all of these carbons left over, and we can get this process to occur over and over. All we've done is lost 2 carbons in the entire process, okay? So the thiolysis is the attack of a molecule of coenzyme A on that beta carbon. And so not only do we produce acetyl coenzyme A, which is going to enter the citric acid cycle, so we're going to harvest even more ATP. Now you've just got this molecule, which is going to undergo beta oxidation again. Right? It's going to do it all over again, and we're going to get a whole bunch more energy from that. So we get 12 ATP molecules for each acetyl coenzyme A that's released during beta oxidation. And if you're unsure about how to count that up, I'll show you how to do that before we're done. So here's an example of palmitic acid, of all the energy that we can obtain from palmitic acid. So what I'd like to do is, instead of me just showing you the slide, I want to show you in detail how the beta oxidation of the palmitic acid molecule works. So I'm going to switch back to a blank document here. Just give me a sec here. Let's see, there should be some room here. Yep, there we go. Okay, so we have palmitic acid, which is a 16 carbon fatty acid. So that's palmitic acid could come from, you know, maybe an animal fat, or, you know, maybe you ate a steak or something. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 carbons in the palmitic acid molecule. So again, this is palmitic acid, which has a total of 16 carbons in the molecule. In the book, they have it written out like this. They just write CH3, and then they put all the methylenes in the chain. There's 14 of them, and then they put the carboxylate like this, right? And I have it written as the protonated form, but if you want to write it as the carboxylate, that's the form that would exist at physiological pH, so there's nothing wrong with that. Well, if you're waiting for me to say, you know, this is the kind of thing that you could be asked about on the final exam, here you go. We have 16 carbons, so it might be a knee-jerk reaction for you to say, well, I'm going to have, if I've got 16 carbons, every turn of beta oxidation removes 2 carbons, 16 divided by 2, it's 8. There you go. I can have 8 cycles of beta oxidation occurring. Well, that's not true, okay? Let me show you quickly. If we were to cut 2 carbons here, right, I have 2 carbons there. So there's 1 cycle of beta oxidation, 2, 3, 4, 5, 6, 7. Even though there's 16 carbons in the molecule, it can only undergo 7 beta oxidation cycles. Okay, and that is important for you to know. All right. So what's going to happen is we're going to activate the palmitic acid in the activation step, which is step 1. I'll just write it over here. This is step 1, which costs 2 ATPs, the value of 2 ATPs. This is our energy investment step, if you will. Then we're going to end up with this molecule. So we're going to have our methyl group. We've got our 14 methylenes. We have our carbonyl, but now it's activated, right? We have the thioester bond, coenzyme A, and now we have our activated fatty acid molecule. Well, if this molecule can undergo 7 cycles, so 7 cycles of beta oxidation, right? How many 2-carbon units am I going to end up with? Well, let's count. I'm going to end up with 1, 2, 3, 4, 5, 6, 7, 8. So again, even though it undergoes 7 beta oxidation cycles, I'm going to end up with 8 molecules of acetyl coenzyme A at the end. So we'll write 8 molecules of acetyl coenzyme A. Oops, my high energy thioester bond, coenzyme A, like that. We've got 8 molecules of acetyl coenzyme A. Well, remember that every time we did the beta oxidation cycle, we produced what? 1 molecule of NADH, and we produced 1 molecule of FADH2. So that means that when this happened, we also made 7 NADH molecules, right, in the 7 cycles of beta oxidation, plus 7 FADH2s. Well, we know that we get 3 ATPs for every 1 NADH molecule, and we get 2 ATPs for every 1 FADH2 molecule. So if we take 7 times 3, that's going to produce what? It's going to give me a total of 21 ATPs, and this is just from the beta oxidation. These 8 molecules haven't even entered the citric acid cycle yet, okay? And we're going to end up with 7 times 2, which is 14 ATPs. Okay, and again, that was just from beta oxidation, right? Steps 2 through 5. Now, the acetyl coenzyme A molecule, and there's 8 of them. Those are all going to enter the citric acid cycle. So I'm just going to write it the way they do it in the book. I'll put it like this, and we'll say this is the citric acid cycle. What do we produce throughout the citric acid cycle? Well, if we have 8 molecules of acetyl coenzyme A, we're going to do the citric acid cycle 8 times, aren't we? 8 times the citric acid cycle. Well, what do we produce in the citric acid cycle? We produce 3 NADHs. So 3 times 8 NADHs. Well, if you take your calculator, so I'm not going to write down all the math right now. Well, I got room. We have 3 times 8 NADHs. And again, you know the value of 1 NADH, which is 3 ATPs. Right, look, NADH cancels, and we end up with what? 3 times 8 times 3, which gives us 72 ATPs. So 72 ATPs. What else do we end up? We also end up forming 1 molecule of FADH2 in the citric acid cycle. So we have 1 times 8 molecules of FADH2, right? Every 1 FADH2 has a value of 2 ATPs. We cancel this out. So we have 1 times 8 times 2. That's easy. That's 16 ATPs. There's one more molecule that's formed in the citric acid cycle, isn't there? We always end up with 1 molecule of GTP. So we have 1 times 8 GTPs, and you know that 1 GTP has the value of 1 ATP, right? It's a ratio of 1 to 1. So that is also pretty simple. So that gives us a total of 8 ATPs here. 8 ATPs. And there you have it, my friend. So let's tally up what we've produced. I'm going to highlight everything in yellow, all the ATPs that we made. We made 21 plus 14 plus 72 plus 16 plus 8. So if we tally all that up, you get 21 plus 14 plus 72 plus 16 plus 8. you end up with 131. So we'll put here total ATP produced. We end up with 131 ATPs. But then remember, in the activation, we ended up investing 2 ATPs in the activation step. So we take 131, we subtract 2 ATPs, and we end up with the total, which is 129 ATPs is what we ended up with from the one molecule of palmitic acid. Again, the major take-home messages, I'm not going to ask you, to add up, 150 ATPs. I'm not concerned about that. What Mr. Dion wants you to understand are two major points, okay? Number one, you count up the total number of carbons in your fatty acid. Here we had a total of 16 carbons. 16 carbons does not mean 8 beta oxidation cycles. It means 7 beta oxidation cycles and 8 acetyl coenzymes are produced. Okay, those are the major take-home messages, and you should know the value of an NADH and the value of an FADH2. And with all that information, you would be able to calculate how much ATP is produced from any fatty acid that you encounter.